Good morning, students. Today we discussed a holiday junction model. It's a topic of recombination occurs in prokaryotes and eukaryotes during the process of meiosis, and it started in genetics. Holiday junction model for MSc previous students. Various models to explain homologous genetic recombination have been proposed based primarily on genetic observation in bacteria and fungi. Now, it is known that some amount of DNA synthesis also takes place during recombination. DNA replication and repair enzymes are present at the time of genetic recombination. In 1964, Robin Holliday proposed a model to explain the molecular process involved during the exchange of DNA between two homologous double-stranded DNA molecules. The key steps of this model are as follows. First of all, pairing or alignment or synapses between two homologous DNA duplexes takes place. Their sequences are perfectly identical except that they may contain small regions of different genes called alleles. Then breaks or nick occur at identical sites in one DNA strand of both homologous DNA duplexes. Precisely at the same point, the broken ends of strands then invade the opposite complementary strands, creating short heteroduplexes regions because of different alleles. This process is called strand invasion. This crossover structure formed is called Holliday junction. Holliday junction was moves in lateral direction. During this process, a DNA strand is progressively exchanged with the DNA strand of the other helix. This lateral migration of Holliday junction is called branch migration. The original base pairs are broken in parental molecules and new base pairs are formed in recombined strands. If the two molecules have alternate alleles AA, BB, CC, then the exchange of DNA strands during branch migration produce double strand regions which are not identical. These mismatched regions are called heteroduplexes. During branch migration, the heteroduplexes region is elongated. Breakage and subsequent reunion lead to formation of this joint molecule composed of four interlocked strands of DNA. The key features of Holliday junction is the cleavage or cutting across the crossover point which resolves or separates the recombined molecules. To expose two cut sides, the Holliday junction is rotated by 180 degree to form a square penile structure. Resolution of Holliday junction occurs by cutting the DNA strands at the site of cross and rejoining them. Resolutions occurs in one of the two ways. This give rise to two classes of DNA products. The cut site 1 cleaves the two strands which were initially broken at the start of recombination process. The resolution produces two non-recombinant molecules as only exchange of alleles has taken place in the middle region of duplex B and B. Thus, a patch of hybrid DNA is formed. These molecules are known as patch products. The cut site 2 cleaves and rejoins two duplexes in such a way that flanking or peripheral genes are exchanged. Here, DNA is reciprocally combined. Crossing over occurs between A and C genes. Now, single strand exchange recombination. According to Masselson and Redding, a single strand break in one of the two homologous DNA molecules is quite common. The free end of the broken strand invades the unbroken double helix and displaces one strand. Rec A protein, or we can say recombination A protein, has the ability to pull out a single strand and displace it. Exonuclease degrades the displaced single strand DNA loop. The unbroken strand of the first molecule serve as a template to synthesize the new strand. Two holiday junctions, double stranded breaks in both strands. Here we discuss about two holiday junctions. So, double strand breaks in both strands of one DNA molecules occur quite frequently during DNA repair of double strand breaks. Homologous recombination occurs. This type of genetic recombination is called double strand break repair mechanisms. These types of breaks may be caused by ionizing radiations and various damaging agents. Here is the diagram. Enzyme RAC or recombination BCD further degrades the broken DNA strands. It generates single-stranded tails with three ends. One 3' end tail invades the other unbroken homologous DNA molecule, displacing one of the strands. The invading 3' end serves as a primer for new DNA synthesis. The displaced strand serves as a template to fill the gap left in the first DNA. If different alleles are present at the site of break, they are permanently lost as regeneration involves homologous DNA as template which has different alleles. This process is called gene conversion because genes of the broken strand are replaced by genes of the homologous DNA. Is the diagram of double strand break repair model with two holiday junction. In E diagram, there is a double stranded break and then double strand break in both strands of DNA molecule at 3' end. Degeneration of broken strands and generation of single strand tails with 3' ends. In next, see, there is a displaced strand, or we can say D loop, and it shows strand invasion and then double strand break repair model with two holiday junctions. In the diagram D, we will see there is a two holiday junction occurs during this process. That's why this process is called two holiday junction model. In double strand break repair, two holiday junctions are created. These holiday junctions move laterally by branch migration and the cleavage resolves and separate the two DNA molecules, forming a crossover and a non-crossover structure. This is similar to the resolution in the single holiday junction. And then enzymes of homologous recombination. There are various proteins that catalyze various steps in the process of homologous recombination in E. coli. Here is the diagram. Enzyme RAC BCD load onto one end of DNA, double strand break and move along DNA recombination. In the process, it unwinds DNA helical and degrades one or both DNA strands. RAC BCD is an endonuclease enzyme. We will see in this diagram this colorful BCD. These are recombinant BCD enzymes which is responsible for the process of the recombination and to end of DNA of double stranded, and it breaks the double stranded DNA and move along DNA for the process of recombination. In the process, it unwinds the DNA helical. All these recombinant enzymes unwinds the DNA helicals and degrades one or both strands. Here we will see the in E position, these recombinant enzymes degrade the both or single DNA strand. Recombinant BCD enzyme shows endonuclease activity and it acts as scissor and form reciprocal break join in G diagram, and in H it shows non-reciprocal break, or we can say BIR, copy of DNA. So this is the function of recombinant enzyme during the process of recombination, and all recombinant A, B, C and D enzymes are followed used in this process of recombination, and Holiday junction is a model which is given by Robin Holiday to explain the junctions formed between during the process of recombination. So these models are known as Holiday junction model, and sometimes there are two Holiday junctions are formed, so then it's called two Holiday junctions. So it's completed now. Thank you, students.